So what we have here is actually the first ever photo taken of DNA. And this was taken using X-ray diffraction. And what happened was you took an X-ray and you shone it over a gel which had DNA fibers in it, and you basically got a diffraction. And that allowed us to sort of zoom in essentially. And that allowed us to determine a lot of things. For example, the structure of the DNA. You could figure out that it was actually a spiral. And this was taken by Rosalind Franklin, and it's the famous photo 51. And this has allowed a lot of other developments in DNA. For example, the actual model of DNA being developed by Francis Crick and James Watson. And these two guys allowed us to actually figure out the structure of this DNA as a helix, a double helix, with the nucleotides actually binding together. And there were a lot of different theories. For example, some people thought there were three strands. Some people thought the nucleotides were on the outside, the backbone was on the inside. But this was the first ever model that actually, and it worked, and actually is what DNA was, and allowed us to actually visualize it. And that allowed us to unlock so many new things. But this was in 1953, and in only half a century, we've still done so much with DNA. In the 1960s, we tried to do something called mutation breeding. And basically, what we did, we took some DNA that was inside of a cell and we just bombarded things with radiation, plants that would be bombarded with radiation. And out of that, we hoped that a lucky occurrence would happen. And what happened was that would rarely happen because when you are turning things on and off, rarely will you ever get something that actually works. Most of the times we would get something that doesn't even function because the DNA is so horribly messed up. Then we got genetic engineering, which happened around the 1960s and 80s, where we actually inserted DNA that we wanted. For example, if we wanted a fish to glow green, we had a gene that we knew could fluoresce, and basically it would be inserted into the cell of, for example, a fish. That's not really what they specifically did back then, but it's sort of an example. But they inserted this gene, but it would be randomly incorporated in the genome. Not very surgical, not very not very efficient. So what we've developed in the 21st century is CRISPR and all these other different nucleases such as zinc finger nucleases and talons. And they allow us to surgically insert DNA, and that's really important. And it not only allows us to surgically insert DNA, but allows us to make small knockouts where we get rid of a gene that we either want to test or know its function. So this is a really big breakthrough, but there have been so many iterations, and one of them is CRISPR Prime, which I'll be talking about later. But first, to understand CRISPR Prime, we're going to have to understand CRISPR, and that goes all the way back to bacteria. Bacteria actually use CRISPR in a different way that we do. Instead of editing DNA and trying to harness and insert genes, what they do is basically they just try to defend themselves against viruses. Because viruses, specifically bacteriophages, are the worst enemy of bacteria because they are so good at killing bacteria, but bacteria need to evolve and they have their own defense mechanism. And essentially, what they do is they just get rid of this viral DNA that they find in their genome. And this may look complex, but don't worry, it's very simple, and I'll explain it all to you right now. Basically, what happens is a virus bacteriophage injects its DNA in the form of just viral DNA, or it could be RNA, but basically what happens is it injects its DNA so that it can use up the bacteria's own machinery so that it can make more of itself and basically hijacks this bacteria cell. Bacteria has its own defense mechanism. Here is its genome and it codes for trace RNA. This is the Cas9 protein that it codes for. And here you have these spacers, and these spacers are actually different snippets of viral DNA that it has acquired after being attacked so many times and surviving. So it takes different bits of viral DNA after it survives, and what it does is it puts it into these little capsules and it's sort of archive. And once a virus invades, it'll manufacture these CRISPR guide RNAs, and it will create a guide RNA after the trace RNA and this CRISPR RNA mixes together. And this allows for tracing, and then this is sort of and this creates a guide RNA. And what happens is it finds a PAM sequence, and then once it sees that, it compares the original CRISPR RNA and against the DNA. And if it finds a match, then it makes a double cut and it breaks down the DNA. This is great. And the second part about why it's so good is, first of all, when it gets rid of the gene, it shuts it down, and that's really good if we want to do something called a gene knockout, which I talked about a little bit earlier, where here you see that it binds and finds a similar, the exact same actually, genetic makeup of the guide RNA. What happens? It makes a snip, and it basically breaks in half. And what happens is the cell's machinery tries to repair, but since it's really bad at this, it often comes up with mistakes, as shown here. And what happens is these mistakes basically they get rid of the gene because even one wrong protein can destroy the whole entire system, and that gets rid of the gene and it silences it. This is not 100%, which is why CRISPR isn't 100%. CRISPR Prime is 100%. But this is way better than just randomly inserting genes or just bombarding things with radiation. And this is another more in-depth look into what it actually does with non-homologous end joining. And what it does is it basically either gets rid of the gene, but also has a chance of fully repairing it. But that usually isn't always the case. Sometimes there's insertion, sometimes there's deletion, and sometimes there's whole entire frame shifts where that's what we kind of want with a knockout. You want to get rid of the whole entire gene. We can also insert our own DNA if we want to. So that's what we do here with homology directed repair. This is the reverse transcriptase, and it's the second part of the CRISPR Prime model. And what it does is it basically turns RNA into DNA, and it can read RNA and change it into its complementary DNA base pair. And that's really important because this is initially found in retroviruses. And what they do is they integrate their RNA into people's genomes. And that allows for basically lifelong viral infection instead of just either killing the victim, the host, or just being stopped outright. And it lives with the body. And what happens and why we use this is basically the use of PEG RNA, which is a modified type of guide RNA. But instead of having a guide, it also has a template that has extra bits that code for a gene that we want. And what happens is the reverse transcriptase adds on to this template, and it creates the complementary base strands. And what happens is now got this overhang. And what happens with this overhang is the body tries to repair itself because it sees there's an overhang and there's also DNA under it. So what it does is it replaces this old DNA with this newer DNA. And then what happens next is these two don't connect with each other, they don't bind. And then the body figures this out too by editing the other side and changing it back into the sort of edited DNA to complement the edited DNA. And now what you have is a fully edited DNA segment. And this is still not 100% because there are problems that come with the body when we're just basically relying on the body to randomly check and randomly make edits. But it is way better than just using normal CRISPR and relying on the body to mess up and just destroy itself essentially and silence and knock out the gene. But this leads us to a bigger question. When we have so much power and we need responsibility, for example, CRISPR was used already to create designer babies. They weren't like modified to be like superhuman or anything. They were modified to be resistant to HIV. And this person that conducted this experiment was arrested because we don't know the full effects of this CRISPR sort of jam or genetic engineering because we still have yet to figure out a lot of things about the genome. Although we may have mapped it out, we are still learning so many more things every single year. What's important to realize is that we're trying to explore and we're trying to use the scientific method, but we have to do so cautiously because every single time in science we're trying to basically peek and open doors slowly and cautiously. But if we open doors suddenly and rapidly, sometimes we can't close that door if it's a path that we don't want to go down. So it's important to keep in mind that safety and caution should also be kept in with fun, the fun creativity.